Welcome to Session 2, the intersection of biology and chemistry for cancer immunotherapy. This session will be moderated by Shi Chen, PhD, Associate Member of the James P. Allison Institute, and Associate Professor of Experimental Therapeutics at MD Anderson Cancer Center. His lab investigates mechanisms of cancer progression and immunotherapy resistance with a focus on ER stress. The keynote address will be given by Carolyn Bertozzi, PhD, Professor of Chemistry at Stanford University. A 2022 Nobel laureate, she pioneered bioorthogonal chemistry to enable molecular imaging and targeted therapies in living systems. Other notable speakers in this session include Marcia Higas, PhD, Professor of Cell Biology at Harvard Medical School, and member of the James P. Allison Institute Scientific Advisory Board. Her work explores how mitochondrial metabolism influences aging, immunity, and cancer. Hojong Yoon, PhD, Assistant Member of the James P. Allison Institute, and Assistant Professor of Experimental Therapeutics at MD Anderson Cancer Center. He develops molecular glues that target proteins previously considered undruggable to reprogram protein interactions in cancer. Gabriel Rabinovich, PhD, Director of the Laboratory of Glycomedicine at the Institute of Biology and Experimental Medicine in Buenos Aires. Dr. Rabinovich discovered galectin-glycan regulatory mechanisms that enable immune escape in cancer. Please welcome Dr. Shi Chen.
Well, thank you for coming to the second session, Interaction of Biology and Chemistry in Cancer Immunology. So integration of biology and chemistry is one of the most exciting frontiers in cancer immunology. We're set to merge about the molecular design with the immune modulation to set for smarter, more effective, and durable response in patients. So today, we have a great honor to have lined up 4 world leaders on chemical biology, cancer metabolism, and cancer immunology to share how to click chemistry to biology and how to glue the sugar and sweetness to immunity. So if you ask me one person who has made the boundary between the biology and chemistry blur in the most effective way and most creative way, I think that person is Dr. Carolyn Bertozzi, who is the winner of the 2022 Nobel Prize in Chemistry. So Dr. Bertozzi invented orthogonal chemistry. I'm sorry for my pronunciation. But that is really revolutionized how we understand about cell surface glycans and open the new frontiers in the drug development, imaging, and the drug delivery. So beyond her pioneering work on the click chemistry, she also clicked music and fun to science. Probably many of you don't know, she was a keyboard player in a band called Board of Education back in 1980s with the famous Grammy winner Tom Morello. So they even won the Ivy League battle of the band in 1986. So I hope over the years, we have no question that Carolyn has already made the education life sport more creative and in a much more fun ways. I think you have inspired all the next generations of scientists with the bold ideas, click the ideas together, which nobody ever thought to do, and that inspire new generations. So without further ado, please join me in welcoming Dr. Carolyn Bertozzi, who is the pioneer who made the chemistry both beautifully precise and scientifically fun.
That's a great introduction. It's a lesson for me that if you're going to coin a term, make it pronounceable. So that's on me. That's not on you. So I love this symposium. I've been so privileged to be here now for the third year. Whenever I'm here in this glorious atrium, I feel like it's a home away from home. This is a community that is so rich in science, in empathy, patient care combined with the leading edge of technology, brilliant people who work selflessly with each other, collaboratively, and the music really rocks. So I have to say, it just checks every single box. So thank you so much to Jim and Pam for including me again in this amazing symposium. I thought what I would do today is talk about kind of the origin story of this unpronounceable brand of chemistry that we call bioorthogonal chemistry, and then end with some of the more modern ways in which these types of chemistries are being used to make next generation bioconjugate therapeutics. So the backstory, which actually touches on another major theme of my research, started with an interest in cell surface glycans. And by the way, I love the speaker lineup in this session because it's a really great mix of chemical biology, therapeutic science, and as you'll see, glycobiology later in the morning. And I started thinking about glycoscience during my years as a PhD student when I was focused on the chemical synthesis of this complex brand of biomolecules. They are found quite prominently on the surfaces of cells, and they come in different shapes and sizes, and you can get from the cartoon here how diverse they can be. But what really struck me was the important role that cell surface glycans play in cell-cell communication, and especially in the context of the immune system. So I shifted from being a carbohydrate synthetic chemist to being more of a glycobiologist during my postdoctoral years in an immunology group at UCSF. And then when I joined the faculty at UC Berkeley back in 1996, the theme of my lab was to develop chemical tools to help advance studies of the biology of cell surface glycans. And one of the interesting discoveries around that time, and this again takes us back 30 years, was how the patterns of cell surface glycosylation change in the context of disease. Specifically, there was already an emerging literature, even back then, around specific phenotypes associated with cancer. So for example, I was reading articles about the upregulation of a particular type of glycan, which we call the sialoglycan. And those are glycans that terminate with this particular building block structure, sialic acid. It's one of the monosaccharides. 2 years ago, when I was at this symposium, I talked about this phenotype. And I analogized to normal healthy cells as having like a well-manicured garden of these sialoglycans, which then transforms into more of a tropical jungle of sialoglycans in the context of cancer. So many cancers have a so-called hypersialylated cell surface glycome. And 2 years ago when I was here, I talked about now in 2025, then it was 2023, what we know about this phenotype, how this relates to cancer immune suppression, and a new type of therapeutic molecule that we've been developing. And I showed a cartoon of a lawnmower mowing these little purple diamonds off the surface of the cell. Who was here for that talk? Or who remembers the lawnmower? Okay, great. I'm not going to talk about that at all today because I already talked about that. But back in the 1990s, when I was just launching my new lab at Berkeley, one of the big frustrations I experienced was that it was really difficult to characterize these kinds of phenotypes in like living systems or in animal models, preclinical models. And I was frustrated because, by contrast, people who were studying kind of every other type of biomolecule had all these great imaging tools at their disposal. So the 1990s was very much like a decade of technology development for molecular imaging. The microscopes were being improved upon at pretty fast clip. People were developing very high resolution, ultimately super resolution techniques for fluorescence imaging in laboratory settings. Clinical diagnostic imaging was taking off. There were new reagents for PET scanning and MRI contrast. And all of these amazing tools were focused pretty much just on two classes of biomolecules, proteins and nucleic acids. So if you studied proteins in the laboratory, you could fuse them to something like the GFP, right? Or M-cherry and all the different colors of these genetically encoded reporters. And that allowed you to visualize your protein of interest in cells, in transgenic animals, and this was just an amazing boom for studies of molecular science in living systems. By contrast, people like me, who wanted to look at the cell surface glycome in living systems, did not have any tools. There weren't tools for imaging glycans. And that was one of the first unmet needs in technology that my very young lab at UC Berkeley tried to address. And so how did we do that? We did it using metabolism and bioorthogonal chemistry, ultimately. And the inspiration for this came from an encounter I had with Professor Werner Reuter. Unfortunately, he passed back in 2016. But in the 1990s, his lab was in its prime, focusing on studies of sialic acid biosynthesis. So I went to a conference, and I happened to hear him present this work back in the early 1990s. What he was working on was basically understanding the enzymes in the pathway that convert a metabolic precursor called N-acetylminosamine. And do I have a laser pointer on this thing? That's not, none of this is a laser pointer. All right. Up there, you see that sugar, it has a pink side chain. That's N-acetylminosamine when the R group is a methyl group. And in our cells, that intermediate gets converted to the corresponding sialic acid, which ends up on the surface of your cell at the terminus of these sialoglycans. And he was studying sialic acid biosynthesis because of the important role that the sugar plays in all these different disease settings, as I was also interested. And what struck me in his talk is that he had mentioned that you can chemically synthesize altered variations of N-acetylminosamine where the R group can be something else besides a methyl group. Nothing too big, but simple 1-carbon extensions all the way out to a 5-carbon chain. And all the enzymes in this pathway would recognize that modified side chain and do the metabolic conversion. So in other words, you could feed cells these chemically altered side chain variants of N-acetylminosamine, and a few hours later, you would see that side chain modification appearing in cell surface sialic acids. So I sat there in the audience, and he showed this slide, and I was like, wow, what if you could use that R group as a place to install a chemical handle, and then use the chemical handle to attach an imaging probe in a biological setting? And a few months later, I sent out applications for faculty positions. This was, in the fall of it must have been fall of 1993, kind of heading into 1994. And I made this cartoon, and I put this in my proposals, and I said, I have an idea for imaging cell surface glycans. We'll start by feeding a cell or an animal a synthetic sugar with a chemical modification, call it X. And the cell will metabolize this precursor and install it within these cell surface glycans. Then, in a second step, we'll introduce a probe molecule, and it will have its own complementary chemical group, let's call it Y, where X and Y have been designed to react very selectively with each other. Now, the chemical reaction would have to occur on the surface of a living cell, and maybe even in a living animal, which means you can't just pull any old chemistry off the shelf. The chemistry has to have very special properties, and there was no chemistry at that time that satisfied these properties. And we ended up defining these properties with this term bioorthogonality. Okay, so here's the definition. Bioorthogonal literally means not interacting or interfering with the biological system. So in other words, for any chemists in the audience, this is a chemical reaction that has to treat cells and animals as if it's like anhydrous THF. Get it? Okay. So another way to put it is there is in your body what I would call the biological reactivity space. So that's all the chemicals in your body. That is a space. It's a big space. You have lots of different chemicals in your body, but there is a boundary around it. Okay, it's not infinite. There's a much larger universe of chemistry outside of the biological setting. And we thought maybe somewhere in the universe there's another galaxy of bioorthogonal reactions. And if we could figure out what they are and import them into the biological space, ideally they would not interact or interfere with that space. So we spent the first few years in my lab at Berkeley, which takes us into the late 1990s, trying to discover the chemistries from that other bioorthogonal space so that we could use them in the biological setting. So I'll shorten the story and get right to the chase, that the first bioorthogonal reaction we developed, which seemed to have many of the qualities we needed for this biological setting, was something we called the Staudinger ligation. We named this chemical reaction after Hermann Staudinger because he was the first chemist to describe the reaction between azides, that's N3, that's a functional group, we call it the azide, with that other group, which is a triphenylphosphine derivative. And azides and phosphines are not in that galaxy of biology. They are in this other galaxy. They are chemicals that humans invented, that people have been using for many applications, but had not been brought into the world of biological exploration. Okay, so we did that, and we named the chemistry after Hermann. Now, by the way, Hermann Staudinger won the Nobel Prize in Chemistry in the 1950s. He first described this reaction in 1919, right? He did not win the Nobel Prize for anything like this. His Nobel Prize was given for his work in polymer science. He was one of the foundational polymer chemists in the world, as a factoid. Now, for reasons I won't go into, we were able to progress this chemistry all the way to studies in animals. You can do that reaction in the body of a mouse with no obvious harmful consequences. But ultimately, for applications in molecular imaging, which was the original goal, this chemistry fell short. For the simple reason that is a relatively slow reaction, takes many hours for these two chemicals to find each other and form a bond. And in living systems, we found that the molecules were just getting metabolized and cleared before they had a chance to react. So in the early 2000s, we kind of shifted our focus towards trying to discover new bioorthogonal reactions that were fast on the timescale of biology, all right? Now, while we were looking for the perfect reaction, two other labs published this chemistry. This is a different reaction with azides, in which the azide is now doing a cycloaddition with an alkyne. That's a triple bond. And in the presence of a copper catalyst, those two will form the triazole product, and it's a pretty fast reaction. This is the chemistry that was invented simultaneously in the labs of Morten Meldal at University of Copenhagen and Barry Sharpless, who's at Scripps. And you all have probably heard of this because this reaction got the brilliant, much more pronounceable brand name of click chemistry. How many people here, especially the biologists, have heard of click chemistry? It might be the only reaction a biologist knows, or has heard of at least. How many people here have actually done a click chemistry reaction in your laboratory? Proudly raise your hands. Yay. Okay, good for you. They shared the Nobel Prize with me in 2022. And by the way, this was Barry Sharpless's second Nobel Prize. He's the only living human on Earth walking around with 2 Nobel Prizes. Okay, so that's an important name to remember for history. Okay, now this is a phenomenal reaction that is now widely used to link things together, click them together in biological settings. But we knew pretty quickly that this would not solve our problem because that copper catalyst is toxic to live cells or live organisms. So those of you who did a click chemistry reaction probably did it on cell lysates, tissue samples, dead cells, recombinant proteins. But if you want to do this on a live cell or in a live animal, kind of dead in the water, all right? Which is why we kept working and ultimately found a solution to the fast reaction problem that didn't require a copper catalyst. And that was basically to put the alkyne into a strained ring system. Alkynes like to be linear. They don't like to have their bonds bent. And if you force them into an 8-membered ring, you got to bend the bonds. And that causes a discomfort in the molecule that gets relieved when it reacts with an azide to form a product. So we call that bottom reaction the strain-promoted azide-alkyne cycloaddition, but even better, copper-free click chemistry. And this was the chemistry that ultimately was the most useful for imaging those cell surface glycans. And we did that. In a typical type of experiment, we would get the azide into the glycan of interest. And of course, this could be broadened really to any biomolecule. So this chemistry can be used for proteins and nucleic acids, anything you want to visualize. But for us, it was all about the glycans. And putting azides into glycans turned out to be the easiest part of this project. We knew from that precedent, from Werner Reuter's work, that an azide on that side chain of N-acetylminosamine should be ignored by the biosynthetic enzymes that convert it to the corresponding sialic acid. And that worked beautifully. And you could put an azide in pretty much any sugar that you want to deliver to the surface of a cell. In humans, we did some work with fucose, which is an interesting sugar for other reasons, with N-acetylgalactosamine. People have now done it in bacterial polysaccharide biosynthetic systems, peptidoglycans, glycolipids. People have done this to put azides into plant cell wall polysaccharides, C. elegans, Drosophila. Okay, so pretty much any living system, you can find a way to get azides into your cell surface glycans, which now arms them for a copper-free click chemistry with an imaging probe. In my lab, we used this concept to do some studies of the dynamics of cell surface glycosylation in developing vertebrates. And the zebrafish is a wonderful model organism for developmental biology. First and foremost, you can form zebrafish embryos in vitro fertilization. And they develop from the single-stage embryo to pretty much an adult fish in 5 days. And because they're optically transparent, you can visualize all of the organ development happening in real time in the live organism if you have a way to get a probe onto your molecule of interest. So we were able to basically develop zebrafish embryos in plates and just dump azido sugars into their media. And those sugars got taken up by the cells and metabolized, and we could then image them on the surface of cells all the way through until the larvae were 5 days old. So that head is a 5-day-old zebrafish where 3 different sugars were labeled with 3 different imaging probes so we could see how they distributed on the tissues as the animal matured. And at earlier time points, so that ball, that's a 24-hour-old embryo. There's a lot of cell division going on. You can watch it in real time. And we noticed some interesting features of the cell surface glycome during these rounds of cell division. So you see those arrows are pointing to cells that have just finished a round of mitosis, where a mother cell divided, formed 2 daughter cells. And we started to notice that a certain type of sugar, a mucin-type glycan, was building up and concentrating right at the junction as the cells were in the later part of the mitotic cycle. And down here is a blow-up of one of those cells undergoing a round of mitosis. And each frame is the same frame. You're looking at the same cell, but we have different labels in each of those frames with different colors. So on the left, that's a fluorescent probe that's on histones. So that's basically like chromatin staining. And you can see classic mitosis, the chromosomes, they duplicate and then they segregate, right? Like right out of the textbook. That's what cell division looks like through the window of chromosomes. Now, one more panel to your right, that's a membrane protein that has a fluorescent probe on it. It's like a GFP fusion. And you can see that the mother cell starts as one cell, and then it starts to divide, and new membrane fills in the junction between the cells. That is what mitosis looks like if you're a membrane protein, okay? Finally, on the third panel, this is like the new revelation. Now you get to see the cell surface glycocalyx during mitosis. And it looks like a fuzzy coating, which you might expect given the structures of these complex polysaccharides. But we notice right at that junction, there's a buildup, a concentration of these mucin glycans that we were staining. And it turns out that they are part of the cell segregation phase. They help push the two cells apart at the end of a round of cell division. And that was kind of a fun observation because you wouldn't know that unless you had a way of casting a spotlight on the glycans. And now that we can see them, we can make observations and try to understand more about the fundamental contribution of cell surface glycans to complex biology, like cell division. Okay, so at some point, kind of in the 2010 period of time, there was a shift in the emphasis in this field of chemical biology from applications of bioorthogonal chemistry in the basic exploration of molecules like glycans over towards the use of these chemistries to make new types of therapeutics. So that's the translational phase of this area of chemical biology. And my lab was getting more and more interested in applications in therapeutic development. And that is what prompted me ultimately to move my lab from Berkeley to Stanford back in 2015. And Stanford is a wonderful environment for translational medicine because just like here at MD Anderson, we've got the hospitals, the physicians, the clinical people on the same campus with the basic science and the technology developers. And I wanted to basically put myself in that kind of an ecosystem, which you can totally understand here at MD Anderson. So fast forward to 2025, and now bioorthogonal and click chemistries are deeply embedded in the biopharma industry, where people use them to make all kinds of creative hybrid therapeutic constructs. Antibody conjugates are by far the leading application right now of bioorthogonal and click chemistries, and mostly antibody drug conjugates, or ADCs. But they're also being used to make vaccine conjugates, cell therapies, oligonucleotide conjugates, basically any setting in which you want to take a large, complicated biomolecule, like a protein or a piece of DNA or RNA, and do chemistry on it. Your first choice is going to be one of these bioorthogonal click chemistries. So I thought I would show you a couple of concrete examples from a world that I'm most familiar with, which is antibody drug conjugates. People here are probably familiar with ADCs. Some of you who are physicians might be treating patients with ADCs. How many people here have a touch point with ADCs? Yeah, okay. And if you don't yet, you will, all right? Because ADCs are still, right now, one of the fastest growing classes of molecules in oncology. And they are exactly what they sound like. They're chemical conjugates between an antibody that targets a tumor antigen and a payload, which is typically a toxic drug. And the logic here is that if you can put your toxin on an antibody and use the antibody to deliver it, then you can get a higher therapeutic window because you're restricting the activity of that toxin to the target tumor cell and ideally keeping it away from healthy cells that you don't want to harm, which causes the side effects of chemotherapy. Now, for the antibody-drug conjugate to work properly, the antigen it binds to has to internalize it so it can get into the endosome-lysosome pathway. And then within that pathway, it gets deconstructed, and that toxic drug gets released from the antibody so that it can kill the cell. And the two mechanisms that are at play right now are toxins that damage DNA or disrupt microtubules so that mitosis cannot occur. And then sometimes the drug is designed so it can get out of the first cell and into a bystander cell and kill surrounding cells, not just the cells that have the antigen of interest. This is an area that I really love because it's such a great example of what can happen when you bring chemistry and biology together, literally. And it's led to the approval by the FDA of now, I think the head count is around 15 ADCs for a variety of indications that target different antigens. There's been a really interesting evolution in this field. It had a bit of a slow start in the 1980s and 90s, but really picked up steam in the 2000s. And there have been various generations of ADCs in which people in the industry have tried to solve the various problems and improve their profiles. So to just summarize where the improvements have focused, you can kind of look at this cartoon, which is like the anatomy of the ADC. And I would say in the 1990s, a lot of the optimization was taking place around the antibody, right? That's the decade where we figured out how to make high affinity, high specificity antibodies, how to get them to express at high levels to improve the cost of goods, how to humanize them or make them human from the first place. And then in the first 2 decades of the 2000s, a lot of the focus was on optimizing the linker and the drug. What's the best type of toxin? How potent does it have to be? The linker, it has to be stable in circulation. You can't just let the drug fall off, but you have to get the drug off inside the cell. And how do you optimize the linker for that paradox? But where bioorthogonal chemistries are coming in is right there, the attachment site. How do you actually form the chemical bond between the drug and linker, which comes from chemical synthesis, and the antibody, which comes out of a cell? And that's where bioorthogonal chemistry is really shining on this industry. So to sort of give you an idea of why bioorthogonal chemistry is so useful and powerful, let's back up and talk about how people have been making ADCs to date without bioorthogonal chemistries. And a good example would be looking at some of the first generation ADCs that got their approval around 2012. So for example, Kadcyla. That was the first ADC targeting HER2. Kadcyla is a conjugate between trastuzumab, which already was an FDA-approved drug, it's Genentech, linked to mitanzine, which is a microtubule disruptor. And the chemistry that connects the two is what I would call old school bioconjugation chemistry. Old school meaning you do the chemistry on one of the natural amino acid side chains, typically lysine or cysteine. And this is a lysine conjugate. And there's the actual chemistry. It's done with an activated NHS ester, as we call it, to form an amide bond on the side chain of those lysines. But here's the problem. Your antibody, like trastuzumab, might have 50 lysines or more on the surface of the protein. And there's no way to distinguish between them when you do a chemical reaction. And if you modified all 50 with this drug linker, you would end up with a garbage pile of precipitate, okay? You have to tune the drug-antibody ratio, which we call the DAR. And the optimal DAR for most antibodies is in the single digits. 2 drugs, 4 drugs, 6 drugs, maybe 8 drugs, okay? So if you're gonna modify 15 lysines and try to get an average of only 4 drugs, you're gonna end up with a lot of heterogeneity in your product. And that is the case for Kadcyla. If you kind of plot the drug-antibody ratio, it's a Gaussian distribution with an average around 3.5. But within that mixture, some of the molecules have 0 drug, some of the molecules have double-digit drug molecules, and everything in between. And you don't know, of all the molecules in your batch of Kadcyla, which is the best. One of those molecules is the optimum ADC, but you'll never find it doing this kind of chemistry, okay? Things get a little better if you use cysteine as your target residue. And what people do is they'll reduce the disulfide bonds at the hinge region of the antibody. There's 4 disulfides on the hinge, so you can reduce them to get 8 cysteine residues. And so when you do the chemistry on cysteine, which is what happens when Seattle Genetics, now Pfizer, makes Adcetris, is you get heterogeneity, but it's not nearly as bad as the lysines, because you don't have 50 cysteines, you only have 8, and you can get a distribution around that 8, which is just a little bit better control, okay? But it's still a distribution with a mixture of molecules where you get a statistical average, but within that mixture, you never can identify the best, okay? Now, if you could do bioorthogonal chemistry, which liberates you from the natural biological functional groups, you, in principle, could have more control over the exact site of modification and the exact stoichiometry. And if you can control your ADC structure and actually do structure-activity relationship, or SAR studies, now, in principle, you can find a best molecule and progress your best molecule instead of a mixture. And so the name of the game here is forget the lysines, forget the cysteines, figure out how to put something new and orthogonal, the X group, into your protein at the site and stoichiometry that you want, and then direct the chemistry precisely to those sites to get an ADC with a homogeneous, defined structure. And there are now several companies and academic groups who have applied various methods to achieve just this, all right? I'll show you the case that's closest to my heart, which is a technology that my lab developed and spun out into a company that was called Redwood Bioscience, but was acquired by Carolyn back in 2014, and this is now a technology within the Carolyn portfolio. What we do is we leverage a natural enzyme called formyl glycine generating enzyme, or FGE for short. FGE will convert a cysteine within a short consensus sequence of 5 amino acids to the corresponding aldehyde, which is the residue formyl glycine. And so when we clone this little sequence into an antibody heavy chain or light chain, that antibody will express, the cysteine gets converted within the cell, and when the cell secretes the antibody, it has aldehyde groups exactly where we designed them to be, based on our cloning. Then we do a bioorthogonal chemistry we developed, which is specific for those aldehyde groups, to have precisely the number of drugs we want on the protein at the sites that we choose. And that's one approach. There are others. Oh, by the way, Catalent now has put molecules into the clinic in the hands of Exelixis with this technology. Okay. I really love what Ambrx is doing. This is a company that spun out from Peter Schultz's lab at Scripps. And they use that Schultz technology of unnatural amino acid mutagenesis, or like genetic recoding. They put a stop codon at the place in the gene that they want to encode a new unnatural amino acid, and then they engineer an aminoacyl tRNA with the unnatural amino acid to match that stop codon. And that has allowed Ambrx to make site-specific antibody-drug conjugates with auristatin analogs that are now in the clinic in the hands of J&J. And then there's a company called Synaffix in Europe, which I love because they have combined some glycobiology with copper-free click chemistry to do site-specific antibody-drug conjugate synthesis, leveraging the conserved glycans that you would find near the hinge region on every IgG. And so they take those glycans, they do some glycoengineering, put in an azide, and then just click the drugs right there onto the glycosite. So that's the ADC example, but there's an example in the cell therapy world too, which is very recent. I don't have to explain to you all about how CAR T therapies work, how transformative they can be, but also how difficult they are to scale. Right now, CAR T therapies require an autologous transplant where the T cells get removed from the patient, genetically engineered ex vivo, and then returned into the patient's body, and then that patient is monitored. But it's hard to scale this, and it's lengthy and it's expensive. So there's a lot of interest in a next generation approach, which would take therapeutic immune cells off the shelf and deliver them to any patient. And there's a company called Asapodia, which has a really clever way of doing this with gamma delta T cells. So my understanding of this is that the gamma delta T cells are less prone to allogeneic rejection and are therefore more useful as off-the-shelf T cells for a broad group of recipients. But they're difficult to genetically engineer with, for example, a CAR T. So instead, what Asapodia does is they take off-the-shelf gamma delta T cells from pooled donors, and then they chemically conjugate them with a targeting antibody that directly reacts with the T cell receptor. And that's their proprietary secret sauce. That allows them to make antibody cell conjugates. So instead of ADCs, these are ACCs that are targeted to a tumor antigen of interest, but off the shelf and not even requiring genetic engineering. And they are now starting to get some early stage clinical data that makes this technology look really attractive to me. But I'm really excited because they were able to do this with bioorthogonal chemistry. So with that, let me leave you with the parting message that bioorthogonal chemistry can open doors towards new types of therapeutic constructs. And that is very much what is kind of the focus of the field right now. But also, I only talked about a handful, not even a handful, of chemical reactions that people are using. But there's still a lot of space to explore that galaxy, to develop new chemical reactions. So bioorthogonal chemistry really has something for everyone. It's got something for biotechnology, something for the biologist, but also some really cool challenges for the next generation of synthetic chemists. And with that, thank you very much for the invitation. I appreciate it.